until now.
Unless you're allergic to micro.
Does everyone have a copy of the study guide?
Yes.
If not, I got some extra copies up
I
here.
don't know if Samantha is, but I'll defer, if you'll send me that.
I thought I sent it. Did I send
You
it?
did.
Okay.
You did. I'm sorry.
No, you're good.
My phone is. I'll try to figure out.
Uh-huh.
So what we're going to do today, we're going to start reviewing chapters 5, 6, 7, and 8. Your test will probably be next Tuesday, so a week from today. I made somebody happy. Yay! That's my goal. Today in lab, you guys are going to do throat cultures on each other. So please bring this handout with you to lab. This pretty much
Dave.
is what we're doing in lab. So definitely bring this handout with you to lab.
I don't feel so savvy today.
Okay. So our computer's taking its sweet time getting ready. I'm going to dive on in. So with chapter 5 is microbial metabolism. You guys remember this chapter?
Yes.
It's a fun one. No, I'm joking. It's not that good, but let me hit the
Appreciate the
highlights
honesty.
of what you should know from this guy. You should definitely know the definition of metabolism and the two major types of reactions: catabolic reactions versus anabolic reactions. So who can tell me what's the definition of metabolism?
Sum of all chemical reactions.
Yeah, it's the summary of all the chemical reactions within a living organism. And that living organism could be a bacteria, it could be a fungus cell, it could be an animal, it could be a person. Doesn't matter. So that's the definition of metabolism. It's a summary of all chemical reactions going on within a living organism. And usually it's over a time period, to be honest. They'll usually say within 24 hours or 48 hours. And when we talk about chemical reactions, there's two major types. There's catabolic reactions and there's anabolic reactions. Who can tell me what is the difference?
Catabolic is the breakdown and anabolic is building back.
Yes, that is correct. So with a catabolic reaction, you're taking a large molecule and breaking it into smaller molecules. So for example, catabolic, you got a big molecule of glycogen. Glycogen is how your body stores glucose. So you have multiple glucose molecules chemically bonded together. So what your body will do when it needs energy, it'll break some of these chemical bonds and release individual glucose molecules into your bloodstream. So that'd be an example of a catabolic reaction. You're taking a larger molecule and breaking it down into smaller molecules. And this, do you give or release energy when you do this? Yeah, big teaching point. You always release energy with a catabolic reaction. Because remember, you're breaking chemical bonds, and chemical bonds are stored energy. So when you break chemical bonds, you're releasing energy. When you make new chemical bonds, that requires energy. So anabolic reactions are the opposite. You're basically taking little molecules and you're making a big molecule. So what's an example of that?
Yeah, the glucose to glycogen.
That's the catabolic. Well, I'll give you an easier one. Take a whole bunch of amino acids. So you have a whole bunch of individual amino acids. And if you start chemically bonding them together, what do you get?
Protein.
A protein, yeah. So you take a bunch of individual amino acids, you're taking a whole bunch of small molecules, and then you chemically bond them all together to form a protein, which is a large molecule. So that's an anabolic. And this requires energy. Why does it require energy? Because you're creating chemical bonds. So if you're creating new chemical bonds, that always requires energy. So that's basically the vast majority of chemical reactions in any living organism. They're either catabolic or anabolic. The other two topics that are really important in Chapter 5, one is enzymes. And we'll talk about enzymes in a little bit. The third one is carbohydrate catabolism. How living organisms break down carbohydrate for what?
Energy.
For energy, specifically to break them down to make ATP. And then I would say lastly, protein catabolism, which is really short. So basically, if you know these 4 topics, you should ace Chapter 5. That's really, I know it's kind of a dense chapter, but when you get to kind of the meat and the potatoes, this is really what Chapter 5 is all about. So this is still warming up. I love it when they do updates first thing in the morning. Like, wait, Dr. Dad was going to class. Hold on, 9:40. Let's do 27 updates. What's an enzyme?
It's a catalyst.
It's an organic catalyst. So what does that mean?
It's the threshold of energy required.
Decreases the.
I think you both were correct. I was just hearing both in stereo. I wasn't quite sure, but yes, you're right. It's basically an organic catalyst. It speeds up a chemical reaction without being used up in the reaction itself. So that's the first definition of what an enzyme is. It's an organic catalyst. And that speeds up the chemical reaction without being used. How does it speed up the chemical reaction, by the way?
It lowers the activation.
Remember that. It speeds up a chemical reaction by lowering the activation energy needed for that reaction to proceed. Every chemical reaction has a cost, if you will. It's basically got what's called activation energy. So, long story short, a enzyme lowers that activation energy so the reaction proceeds much faster. It's like if you're trying to sell a book for $100, you probably wouldn't get a lot of takers. If you wanted to sell the same book for $5, you'd probably sell a whole bunch real quick. So, once again, lower the cost and the results proceed much faster. What actually is the physical definition of an enzyme? It is what type of macromolecule?
Protein.
It's a globular protein. So remember, enzymes, besides being organic catalysts, are also globular proteins. They're basically spear-shaped proteins. Now, important teaching question. Do enzymes work all by themselves? What do they need to become activated? Well, that's what they work on, but how do they get activated before they can even work on a substrate? A cofactor, yes. They must have a cofactor to basically turn on. Okay. Now that the computer's finally on. Yeah, an enzyme by itself does nothing. Think of it like your car without your key or your fob. The car does nothing. But if you have the cofactor, which is essentially the key, then the enzyme becomes activated. Okay. I'll just caught up real quick here. So once again, metabolism is the summary of all biochemical reactions within a living organism. And they're either catabolic or anabolic. Catabolic, you're breaking large molecules into smaller molecules and releasing energy. Anabolic's the opposite, just like we were talking about. You take multiple small molecules, you chemically bond them together, and that requires energy. Okay. And enzymes are organic catalysts. There's several different categories. I'm not going to have you guys remember all the different categories. I think that's not appropriate. But here's what I was getting at. An enzyme with no cofactor is called an apoenzyme. An apoenzyme is a deactivated enzyme. So think of it a car without a key. Once you add that cofactor, then that enzyme gets activated. And that's called a holoenzyme. So a holoenzyme is like your car with the engine running, basically. It's ready to go to work. Why does your body selectively turn off and on enzymes?
To stop the
Well,
reaction?
yeah, to conserve energy, because you don't want all your enzymes working all the time. You know, you have certain times where you want one class to work, different times you want a different class to work. So you want to kind of selectively turn them on and turn them off depending on your body's needs. So that's why a cofactor is necessary. Now, what is a cofactor? Well, it's either one or two things. You guys tell me, what's a cofactor? It's either a... What's that?
No, never mind.
Everyone's forgotten that second.
Yeah,
okay,
I'm
that's
sorry.
fine. It's either one or two. It's either a metal ion or it's a B vitamin derivative, basically. That's the easy answer. So it's either a metal ion. It's not just magnesium, though. It could be copper. It could be zinc. Most importantly, it could be iron. That's the chemical symbol for iron. Yeah, so magnesium, copper, zinc, iron are all cofactors.
Oh, that's the one you said, that's why we take the
Multivitamins, yes, that's why you take vitamins. Vitamins are just a whole bunch of cofactors. So you're basically giving your enzymes lots of fuel, if you will. And then the rest of them are basically B vitamin derivatives. Some of them come from niacin, which is B3. Others come from riboflavin, which is B2. You got B9, B5, B6. This is the one that supposedly cures cancer, according to RFK Jr. Don't know if that's true, but that's what my wife's making me take that now. I'm like, oh God, I hate it. The pills are like that. And I'm like, I'm going to choke to death. And then there's B1.
You won't have cancer?
That's true.
No, that's true. Gotta always look at the
always.
positive side of things. I really think they're like horse pills. Anyways, so that's what a cofactor is. It's either a metal ion like magnesium, copper, zinc, or iron, or it's a B vitamin derivative. And once again, if you look at a multivitamin, that's mostly what they are. They're mostly got magnesium, copper, zinc, iron, a whole bunch of B vitamins, and they'll have vitamin A and K and C, the rest of them in there. But this is basically your cofactors. This is basically what turns your enzymes on so they go work. And this, once again, is kind of the chart that shows you how enzymes actually work. They work by lowering the activation energy needed for a reaction to proceed. So this dotted line is how much energy it would cost without the enzyme. This solid line is how much energy it costs with the enzyme. Obviously, with the enzymes, a lot less, so this reaction is most likely going to happen. It's going to happen very quickly. Why are enzymes essential for living organisms?
We wouldn't be able to produce enough energy to continue it on.
Well, that's actually, I'll say 55% correct. Basically, your body goes through hundreds of thousands, if not millions, of chemical reactions each day. But you want to maintain your homeostasis. And if you remember from AMP1, you don't want your body temperature going above 98.6 degrees Fahrenheit. You don't want your blood pH varying from 7.4. So enzymes allow you to speed up chemical reactions without offsetting your homeostasis. Because in the chemistry lab, how do you heat things up? Whoops, I just gave it away. How do you speed up chemical reactions? You heat things up. You know, you put a beaker over a Bunsen burner, and that speeds up the chemical reactions. Well, you can't throw yourself in front of a giant Bunsen burner, obviously. So enzymes allow living organisms to kind of internally speed up their chemical reactions without upsetting their homeostasis. That's the key. So that's why enzymes are, you know, essential for life, if you will. Does that make sense? Okay. And the substrate is just whatever the enzyme works on. So the substrate, it could be a glucose molecule, it could be an amino acid, it could be a lipid, doesn't matter. Whatever this particular enzyme works on is called the substrate. Now, where does the substrate bind to on the enzyme? Yeah, it binds to what's called the active site. So the active site is the location specifically where this substrate will bind to the enzyme. It forms the substrate enzyme complex. And then the enzyme's going to do what it's going to do. And to be honest, it's usually either going to break chemical bonds or make chemical bonds, depending on the enzyme. So in this example, the substrate is fructose. Fructose is a sugar. It's a cousin of glucose, basically. It binds to the active site of the enzyme. The enzyme splits it in half, and it releases two new products. And then now it opens up, it's ready for business for the next fructose molecule. So that's basically how enzymes work. They're very sensitive to temperature, pH, etc. So obviously they work best at 37 degrees Celsius, which is 98.6 degrees Fahrenheit in the human body. And they need the right pH, which is about 7.4, etc., etc. Now, what's the allosteric site? The allosteric site is what binds to the enzyme. We were just talking about them about 5 minutes ago. Nope, your cofactors. Yeah, your substrate binds to the active site, but before that happens, you have to have the cofactor bind to the allosteric site. Once again, think of the enzyme as a car. It doesn't do any good if you don't have the key or the fob or whatever. And the cofactor, which is this green triangle here, is the key. So you've got to put the cofactor in the allosteric site. Look what happens to the active site. It actually becomes functional. So the active site without the cofactor isn't even the right shape. It's not going to do anything. But once you add that cofactor, that active site actually changes shape to fit the substrate and becomes functional. Now it's ready to go to work. So that's the key difference. Remember, the substrate, whatever the enzyme's working on, binds to the active site. The cofactor, which you need to turn on the enzyme, binds to the allosteric site. Does that make sense? Yes? Okay. Yeah, big distinction here. And that's how you go with competitive inhibitors versus non-competitive inhibitors. Who can tell me what's the difference between a competitive inhibitor and a non-competitive inhibitor?
the active site?
Which one?
The competitor blocks.
You're
the
right,
active-
yes.
Okay.
So a competitive inhibitor is a molecule that blocks the active site. And if it blocks the active site, then the enzyme can't work on a substrate. And we call it a competitive inhibitor because it competes with the substrate for the active site. That's why it's called a competitive inhibitor. Sometimes your body will use these to shut down enzymes temporarily. So sometimes your body will decide, you know what, we don't really want to use this enzyme, we don't really need it, it's a waste of energy, so we're just going to shut it down with this competitive inhibitor, and then later, okay, now we need it, then it'll release the competitive inhibitor. So most of these are reversible, if you will.
Is this the one you said the poison's blocked on time?
Well, a poison would be a permanent inhibitor. So yes, you could have a competitive inhibitor that permanently shuts down the enzyme, and that's bad. So that's what some toxins and some poisons will do. Now, what's a non-competitive inhibitor do?
And you block the cofactor site.
Yeah, it blocks the allosteric site. So if you block the allosteric site, what's that mean? It means the cofactor can't bind. So basically, if the cofactor can't bind to the enzyme is never activated. It's basically shut down. So that's the difference between a competitive inhibitor versus a non-competitive inhibitor. Okay. Now, before we jump into carbohydrate catabolism, which I know is everybody's favorite topic, I want to briefly touch on oxidation reduction reactions. So this will just take a moment. Does anyone remember what oxidation reduction actions are?
Gives and reduced gain.
Very good, yes. This is basically when you're passing electrons from one molecule to the next. And this is what happens in the electron transport chain that we're going to talk about in a little bit. So you have one molecule here that's giving up an electron, and you have another molecule here that's receiving an electron. So we always say the molecule that gives up the electron has been oxidized, and the molecule that receives the electron has been reduced. And the reason we call this reduced is, what charge does an electron have?
Negative.
It's negative. So if this molecule accepts a negative electron, its overall charge is reduced or decreased. So it goes from being more positive to being more negative. So that's where the term reduced comes from. Does anyone remember why we call this molecule oxidized? The one that gave up the electron? Because usually it's giving it up to oxygen. I would say 80% of the time the molecule it's giving up its electron to is oxygen. Oxygen loves electrons. That's what causes rust, by the way. If you leave iron out in the environment and it gets wet, oxygen in the air will eventually take away its electrons, and that's what causes rust, basically. So, all right, so that's oxidation reduction reactions. Okay. Now we're ready to jump into this. How a living organism breaks down glucose to make ATP for energy. Now, what is glucose? We'll start with that. It's a sugar, and it's the most common sugar in the world, and almost every living organism loves glucose to make ATP. Bacteria love it, fungi love it, plants love it, animals love it, and we love it. It's basically glucose is your blood sugar. Now, how do you take a molecule of glucose and make ATP from it? Well, there's a couple ways. Every living organism does what's called glycolysis. You guys tell me, what's glycolysis?
Break it down
Process
into.
that always occurs in science.
Very good. It's a 10-step process where you take one glucose molecule and you divide it into two what we call pyruvic acid molecules. And this always happens in the cytoplasm of the cell. Very important teaching point. Doesn't matter if it's a prokaryote, like a bacteria, or a eukaryote, like a human cell. This always happens in the cytoplasm of the cell. And you also make 2 ATP. So you took glucose, you went through 10 steps, you split it in half basically, and make 2 pyruvic acid molecules. And then you also make 2 ATP that can be used for energy. And that's not a whole lot of energy, but 2 ATP is better than nothing. The trick is, where does the organism go from here? It can either go through the oxidative cellular respiration pathway, which would include the Krebs cycle and the electron transport chain, or it could go through the anaerobic pathway, which would usually be fermentation. So what decides which pathway the organism goes?
Which one uses oxygen?
Yeah, which one uses oxygen? If it uses oxygen, it's going to go through the oxidative cellular respiration pathway, meaning these 2 pyruvic acid molecules are going to go through the Krebs cycle, and then they're going to go through the electron transport chain. By the way, of all this, where do they make the most ATP?
Electron transport chain.
The electron transport chain. So remember, this electron transport chain is where they're going to make 95% of their ATP. We'll talk about that in a moment. If the organism does not use oxygen, think of a lot of fungi that grow in the soil, or even some bacteria like Clostridium or Bacillus. They don't necessarily use oxygen because they live in the soil. There's not much oxygen in the soil. They're likely going to go the anaerobic pathway. Anaerobic means without oxygen, and that's usually fermentation, which we'll talk about. Okay? Everyone with me so far? Okay. So let's do the oxidative pathway first. We'll just say this is an organism that uses oxygen. So you've already gone through glycolysis. You've already gone through the 10 steps, and you ended up with 2 pyruvic acid molecules, and you made 2 ATP. Now, the next step is you want to get to the Krebs cycle. But what has to happen before you can start the Krebs cycle? You got to convert that pyruvic acid to what? Acetyl-CoA. So that's step 1. You got to take those 2 pyruvic acid molecules you got from glycolysis, and you have to convert them to acetyl-CoA. So that's what you do. You take pyruvic acid. There's actually 2 molecules of pyruvic acid that you got from glycolysis, and you convert it to what's known as acetyl-CoA. Once you've got acetyl-CoA, then you can actually start the Krebs cycle. The Krebs cycle is also called the citric acid cycle. Don't get confused by that. It's the same thing. Basically, it's just interchangeable. So with the Krebs cycle, there's actually 8 reactions. And the first reaction is you take the acetyl-CoA and you actually turn it into citric acid. So that's why it's sometimes called the citric acid cycle. And I'm not going to have you guys memorize all 8 steps, because that's beyond the scope of this class. But the long story short is, here's what you get at the end of the Krebs cycles. You get 2 molecules of ATP, which could be used for energy. You get 4 molecules of carbon dioxide, which are waste products. But here's the real benefit. You get basically 8 electron transport carriers. You get 2 molecules of FADH2, which is an electron transport carrier. And you get 6 molecules of NADH, which is also an electron transport carrier. And where do these 8 electron transport carriers go? To the electron transport chain. Thank you. So think of this as getting the fuel ready for the electron transport chain so it can go to work, if you will.
You said there was 8 FADH?
There's 2 FADH2s and 6 NADHs.
Okay.
So a total of 8 electron transport carriers. Now, important teaching point before we mosey onward. Where does the Krebs cycle occur in a prokaryote? In the cytoplasm. Where does it occur in a eukaryote? The mitochondria. Write that down if you don't know it. That's really important. So, why glycolysis is always in the cytoplasm no matter what, the Krebs cycle occurs in the cytoplasm in a prokaryote, like a bacteria cell, but it occurs in the mitochondria of a eukaryotic cell. So, two very different locations. Now, let's move on to electron transport chain. Electron transport chain is where, the organism will make the majority of its ATP. And it's basically a series of carrier molecules, mostly proteins, that basically pass electrons from one to another. So it's basically a series of oxidative reduction reactions until you get to the final electron acceptor. And what is usually the final electron acceptor? Oxygen. So 9 times out of 10, the final electron acceptor is usually a big old molecule of oxygen. Now, where do you find the electron transport chain in a prokaryote?
Plasma membrane.
It's going to be part of the plasma membrane. Where do you find the electron transport chain in a eukaryote?
Mitochondria.
In the mitochondria. So that's another important distinction. So here's what basically happens. Remember all those electron transport carriers we got from the Krebs cycle? The NADHs and the FADH2s? Well, this is where they go to work. They basically come to the electron transport chain and they split their hydrogen atoms. And what happens is, remember, hydrogen is just one proton and one electron. There's no neutrons. So the electrons become part of the electron transport chain. And they just kind of keep going down this chain. What happens to the protons? They get kicked outside the membrane and form a concentration gradient. So you've got this huge concentration gradient of hydrogen ions or protons outside the membrane, like this. Meanwhile, the electrons are just kind of flowing down the electron transport chain because each molecule has a higher affinity than the preceding molecule. So this guy has a higher affinity for electrons than this guy. This one has a higher affinity. This one has a higher affinity. They keep going down until they get to the final electron acceptor. And even though they don't show it here, that's usually oxygen. And when you have a flow of electrons, what's that called? That's electricity, basically. You're not using copper wire, though. You're using a basically plasma membrane or a membrane within the mitochondria. But here's how this makes ATP. Remember, you have all these hydrogen ions outside the membrane that are getting all bunched up together in high concentration. Are they happy being bunched up? What's the natural state with most molecules? Most molecules flow down the concentration gradient. So what's that mean? They like to go from areas of high concentration to areas of low concentration. So what happens is these guys want to sneak back into the cell or the mitochondria to go down their concentration gradient. And there just happens to be an enzyme that has a channel that lets them do that. And that enzyme is called ATP synthase. So with the name ATP synthase, what do you think it does?
Makes ATP.
It makes ATP. So every time two of these protons come back in, it takes ADP and phosphate and ding, makes ATP molecules. Then two more come in, ding, it makes another ATP. Then two more come in, ding, it makes another ATP. So this ATP synthase at the end of the electron transport chain is basically just a little ATP making factory, if you will. And long story short, from one glucose molecule, the electron transport chain can make anywhere from basically 32 to 34 ATP. And that's pretty impressive. So from one glucose molecule. So this is where, for organisms that use oxygen, this is where they make the majority of their ATP, in the electron transport chain. And just remember, in a prokaryote, like a bacteria, this is going to be part of the plasma membrane. In a eukaryote, like one of our cells, this is going to be within the mitochondria. Okay, does this make sense? This is also called chemioosmosis. So if you ever see the term chemioosmosis on, I don't know, a test or something, don't get confused. This means the same thing as the electron transport chain. So chemioosmosis, electron transport chain are the same thing. Okay? Does this make sense? Okay, any questions? Because I know it's a lot of info. No? Okie dokie. And yes, this is where a lot of poisons work as well, is the electron transport chain. This is where cyanide works. It basically blocks one of these electron acceptors. This is where arsenic works. It blocks a different, basically, electron acceptor. So basically, these poisons work by shutting down the electron transport chain, so your cells can't make ATP. If you can't make ATP, you can't really do a whole lot. So that's how they work. And once again, chemioosmosis is just another term of using the electron transport chain. So if you look for a prokaryote that uses oxygen, they can take one molecule of glucose and make a total of 38 ATP molecules. They make a net total of 2 from glycolysis, another 2 from the Krebs cycle, and then 34 from the electron transport chain. So when you add all that up, it comes up to be 38 ATP molecules. Now, eukaryotes make 36 ATP molecules per one molecule of glucose. Why do we make 36? Because it costs 2 ATP to send acetyl-CoA into the mitochondria. Since eukaryotes use the mitochondria, it costs them 2 ATP to transport acetyl-CoA into the mitochondria, so you can start the Krebs cycle and then the electron transport chain. So for a eukaryotic cell, it'd be a total of 36 ATP per glucose molecule, which is still pretty good. Now, what if you don't use oxygen? Yeah, you use an anaerobic pathway. Anaerobic means without oxygen, and the most common anaerobic pathway by far is fermentation. So this is typically used by fungi like yeast. This is also used by some anaerobic bacteria. So you guys tell me, what is fermentation?
It makes ethanol, glucose to ethanol.
Yeah, you basically go through glycolysis, and then you take that pyruvic acid and you turn it into ethanol, which is what? Ethanol is alcohol. Yeah. Wouldn't be drinking alcohol per se, but sometimes it is. So ethanol is just alcohol. And as a result of this, you also make a lot of carbon dioxide gas. That's why beer, and like champagne have bubbles and stuff like that. So you're taking pyruvic acid, and you're converting it to ethanol or alcohol. Well, how does this help, say, the fungus to make ATP? Well, fermentation does not make ATP by itself, but glycolysis does. Remember, you make 2 ATP every time you go through glycolysis. So what fermentation does, it makes some of the precursors that are necessary for glycolysis. It makes NAD+, and this goes back up here and kind of restarts glycolysis. So basically the fungus cell's like, okay, let's burn up another piece of sugar and turn it into alcohol, and it does. And when it does that, it makes another 2 ATP. And then it's like, okay, that was fun, let's do that again. Let's burn up another piece of sugar. And basically fermentation basically motivates the fungus cell or the bacteria cell to basically metabolize all the sugar it's got to make ATP. And that will send it into the fermentation pathway. That's why, if you want to basically make wine, what do? You take grape juice, you throw in a bunch of yeast, and you throw in a bunch of sugar. And because of the fermentation pathway, that yeast will keep making alcohol until it runs out of sugar, basically. All right, so that's basically how fermentation works. It basically re-stimulates glycolysis to happen until you run out of sugar. And each time you're doing glycolysis, you make 2 ATP. Now, ATP, once again, isn't a lot, but, fungi really don't need a lot of energy. You don't see mushrooms dancing and running around, so, they're okay with only making 2 ATP per glucose. So that is how fermentation works. Questions? No? Okay. And this is what I was talking about a couple minutes ago. So with the aerobic pathway, where you go through the Krebs cycle and the electron transport chain, 1 molecule of glucose can be turned into 38 molecules of ATP in a prokaryote, 36 molecules of ATP in a eukaryote. And once again, the reason it's 2 less in eukaryote is we have to use 2 ATP to get acetyl-CoA into the mitochondria so you can start the Krebs cycle and the electron transport chain. With fermentation, which is the most common anaerobic pathway, you basically make 2 ATP per glucose molecule, and that's just through glycolysis, basically. Okay. That is carbohydrate catabolism. Now, the only one you need to know here is protein. How do our bodies break down proteins? Well, there's 2 ways. There's 2 steps, basically. Can a cell just swallow a big old protein?
No.
No. So what do cells have to do before they can get the amino acids?
Break it down.
Yeah, they've got to break down the polypeptide. They got to basically break down the protein into individual amino acids. And that's through the use of enzymes. And we call these protease enzymes. Protease enzymes are enzymes that break down proteins. They break the peptide bonds and they release the individual amino acids. And this can happen outside the cell. That's like why staph and strep release exotoxins. Remember, exotoxins usually are digestive enzymes that break down proteins. Or in our case, when you eat protein, you basically have hydrochloric acid start to break it up in your stomach, and then you have pepsin, which is a protease that kind of finishes the job. But regardless of what type of cell you're talking about, the first step is you've got to break up the protein into its amino acids. Then you can absorb the amino acids, kind of like a milkshake. But once you absorb amino acid, what's the second step? It's really important.
Deamination.
Deamination. What's that mean?
Remove an amine group.
Yeah, you got to get rid of the amine group. So step 1 is you got to break down the protein into amino acids that can be absorbed by the cell. And that's usually done with the protease enzyme. Step 2 is called deamination. Deamination literally means you remove the amine group from the amino acid. Amine is nitrogen plus 2 oxygen, so NH2. You're going to get rid of the amine group as a waste product, because nitrogen's actually very toxic to most living organisms. So the rest of it, you'll use for the citric acid cycle and for metabolism, but that amine group's going to be removed as a waste product. In humans, your kidneys filter out the nitrogen-based waste products. All right, so that's step 2. So first step is you got to break up that protein into amino acids that can be absorbed by the cell. Second step is deamination. Once you get those amino acids in the cell, you've got to get rid of the amine group and secrete it as a waste product. And that's how living organisms digest proteins. Make sense? Okay. And if you understand that, then that is it for chapter 5. So, woohoo! Yeah, that's the most dense chapter I think we got. So, with the time we have remaining, I'm going to go over the high points of chapter 6. Chapter 6 is actually pretty basic. What the big take-home point with chapter 6 is, how do you keep microorganisms alive in the lab? Remember, it's a little opposite thinking, because up to this point, when we talk about antibiotics and antimicrobial drugs and disinfectants and all that, we're talking about how to kill microorganisms. Well, this is like, how do you keep microorganisms alive in the lab for beneficial purposes, to make an antibiotic or to make a product that's going to help a human. And there's some basic concepts. So to keep microorganisms alive in the lab for beneficial purposes, what do they need?
Nutrition.
Yes. So the first thing they need are nutrients. Pretty much every living thing needs glucose and amino acids, etc. And most of that, honestly, is in the soy agar you grow them in. But they need nutrients. What else do they need?
The right pH.
The correct amount of oxygen. Because remember, some microorganisms love oxygen, some hate oxygen. You need the correct pH in the environment. Most organisms do better at a neutral pH around 7. Fungi is the exception to that rule. Fungi likes acidic pHs like 5 or 6. What else do you need?
Temperature.
Temperature. Remember why we have 2 incubators in lab? Because some microorganisms grow best at 37 degrees Celsius, which is human body temperature. Others grow better at 30 degrees Celsius, which is environmental temperature. So you have to have the correct temperature. And then finally, I would say you have to have the correct osmolarity. That's basically kind of a salt to water ratio. Basically, most organisms grow best in an isotonic environment. Okay, so these are the big 5. If you're going to grow microorganisms in the lab for beneficial purposes, you got to make sure they have nutrients. You got to make sure they're getting the right amount of oxygen. You got to make sure they got the right pH, the correct temperature, and the correct osmolarity, meaning they're growing up in an isotonic environment. A lot of these are taken care of by the soy agar. Soy agar has nutrients, it's the right pH, it's the right osmolarity. But it's kind of your responsibility to make sure you put them in the right temperature, i.e. the right incubator. And then oxygen, when I put those little test tubes of bacteria, the cultures, it'll say either open lid or loose lid or tight lid. Basically, if it's an organism that uses oxygen, you actually loosen the lid so it can get oxygen in the test tube. If it's an anaerobe, it'll say tight lid. That means you tighten the lid so oxygen can't get in there. So that's how you do the correct amount of oxygen. So this is one of the big take-homes from Chapter 6. If you understand this, this is like half the chapter of Chapter 6 right there. Okay? So with that, let me go through the specifics. All right, once again, everything must have nutrients. And you must have the right oxygen. So what type of organisms do we have? Well, we've got obligate aerobes. What's that mean? They
have to
must
have-
have oxygen. If they don't have oxygen, they will die. And this is that liquid thioglycolate lab we used to do, is where you put basically bacteria into a liquid culture in a test tube. And the only oxygen's right up here at the top. So if you have an aerobe that needs oxygen, where do they cluster? Right up here next to the oxygen. They all want to get to this oxygen. By comparison, what about an obligate anaerobe?
Oxygen kills them.
Yeah, oxygen will actually kill these guys. These are like bacteria you find deep in the soil. So if you put them in this tube, where are they going to cluster?
Bottom.
Way far away. They want to get as far away from that oxygen as they can, because they know that oxygen will kill them. A facultative anaerobe can go either way. They're an organism that grows better with oxygen, but can also live without it. So I like to say facultative anaerobes, they go either way. If oxygen's available, they prefer to use oxygen because they make more ATP. But worst case scenario, if there's no oxygen, they'll switch to basically fermentation mode, if you will. They'll switch to anaerobic. So as you can see here, most of them will cluster up towards the oxygen, but some of them are like, nah, we'll just use fermentation, we're good. So that's a facultative anaerobe. They can go either way. What about an aerotolerant anaerobe?
Doesn't need it.
Yeah, they don't really care about the oxygen. They're not affected by it, but they also don't use it. So they tolerate oxygen, but they don't use it. So you see, they're pretty evenly spread out through the liquid. They're just kind of ignoring the oxygen up here. Like, okay, that doesn't really affect us. Doesn't really help us, but doesn't hurt us. So they're just kind of spread out. Now, there's a fifth category they don't show up here, and that would be the microaerophile. Who remembers what a microaerophile is?
They like low concentrations.
They just like little tiny concentrations of oxygen. What's the classic example? H. pylori. Helicobacter pylori, better known as H. pylori. That's the bacteria that can get in your stomach and cause stomach ulcers. Don't ask me why, but that bacteria loves low levels of oxygen. In your stomach, you've only got like 10% oxygen. So that's like perfect for a microaerophile. And that's why they like to get into the stomach and create havoc, basically. All right, so those are the 5 categories of oxygen requirements you should be familiar with. Okay. So we talked about nutrition, we've talked about oxygen, temperature. Once again, this is why we have 2 different incubators in labs. Some organisms grow best at human body temperature, which would be 37 degrees Celsius. Others grow best in environmental temperature, which is 30 degrees Celsius. And that's what this is showing here. So kind of, for most organisms, 37 degrees Celsius is preferable. Unless you're talking about something that's purely environmental, then 30 degrees Celsius would be the preferred temperature. pH. Most microorganisms are what we call nutrophiles. They like a neutral pH. So 7.4, which is the pH of human blood, is perfect. What's the exception to that rule?
Fungi.
Fungi, they are acidophiles. They actually grow best in slightly acidic pHs. So let me show you. They have a picture of that later on. Oh, come on. How much farther did they put it? Okay, yeah, they put it about 9 slides ahead because, that's what they do. But here's bacteria colonies, and they're growing well at a pH about 7.3. Look at the fungus colonies. They thrive at an acidic pH of 5.6. Now, you can grow some fungus at 7.3, but it really will take off if you have a slightly acidic pH. That's why, depending on what you're growing, the pH will matter. Okay. So we've talked about pH. Of course, I got to say the exception to this rule would be the arch area. Because remember, arch area are the extremophiles. They love either really hot or really cold, really acidic or really basic. They're kind of the oddballs of the group. But to be honest, we don't really culture arch area. They don't have any really known benefit at this point. Osmolarity. Basically, you want the right salt-water ratio. Because if you put most cells in a hypertonic solution, what's going to happen?
Shrink.
They're going to shrivel up because all the water is going to leave them. They're going to shrivel up like a prune and die. Likewise, if you put most cells in a hypotonic environment, what's going to happen?
Swell up.
Yeah, they're going to swell up from all the water rushing into the cell and basically explode. You don't want that either. So that's why you want basically an isotonic environment when you're trying to grow microorganisms, where the salt-water ratio is the same in the environment as it is in the cytoplasm of the cell. And also, this is what we do with the soy agar. The soy agar is designed to have the same, basically, osmotic pressure that would be inside the cell. So it's basically an isotonic environment. Okay. What's a biofilm?
Block party of microorganisms.
Yes. A block party of microorganisms, yeah. This is where you have several different types of bacteria kind of living in a big commune on a warm, moist surface. So that's what a biofilm is. So it's numerous microorganisms on a surface, and this could be like mildew on a shower curtain. This could be plaque on somebody's teeth. Doesn't really matter. So biofilm is just multiple microorganisms hanging out on a warm, moist surface that they can grow on. Now, if you're growing bacteria colonies, they'll all have different shapes. And when you look at a colony, you're not looking at one individual bacteria cell. You're looking at several thousand bacteria cells kind of lumped together. And sometimes the shape and the color of the colony can give you a clue to what you're growing. For example, with the skin cultures, what was a white colony typically?
Staph epidermidis.
Staph epidermidis. What was a golden yellow colony?
Staph
Staph aureus. That would be one example of that. Okay. Now, what's a pure culture? You guys remember this definition?
It's got a dominant.
Yeah, it's a colony that arises from a single dominant microorganism. I'll say that again. A pure culture is a colony of bacteria that comes from a single dominant microorganism. And what's the best way to isolate a pure culture? It's known as streak plating. And that's where you'll take a sample of the bacteria from the source and you'll make a streak. And then you'll sterilize your inoculation loop. You won't go back to the original source, but you'll take a sample from the first streak and make a second streak. And then you take a sample from the second streak and make a third, And then if you want, you can take a sample from the third streak, make a fourth. Basically, each time you're doing this, you're diluting the sample until by the time you get to that fourth streak, you pretty much isolated a pure dominant microorganism. And this would be your pure culture here. So each one of these individual colonies arises from a single dominant microorganism. So that's called obtaining a pure culture. And streak plating is the most common method to do that. Okay. Now we've already talked about how they make basically growth mediums. You take a nutrient broth like soybean broth and you add agar. What's agar? Yeah, it's a carbohydrate or sugar you get from seaweed. So you add them both, soybean broth, and agar, you get soy agar. And that's the majority of Petri dishes used in lab. However, there is an exception. This literally rolls into lab today. What's a differential medium?
Sheep's growth plate to distinguish between different organisms.
Yes, what she said. It's a special Petri dish that's used to distinguish different microorganisms. That's why it's called a differential medium. It's a growth medium where different bacteria will have different colored colonies. So with blood agar, what is blood agar, by the way? Yeah, it's basically soy agar with 5% sheep's blood added to it. And that's why it looks red. And basically, when you streak different microorganisms on it, you'll get different colored colonies. And with beta hemolysis, you'll get golden yellow colonies. With alpha hemolysis, you get green or brown or silver colonies. And with gamma hemolysis, you get no colonies. What is based on this? The hemolysis in this case is what?
The hemoglobin from the red blood cells.
Yeah, what percentage of red blood cells get destroyed?
100%.
In beta hemolysis. So in beta hemolysis, and I was so nice, I wrote all this down on your handout today, so you guys didn't have to just scramble and take notes. So, yeah, beta hemolysis means the microorganism you streaked killed 100% of red blood cells. And all that hemoglobin from the dying red blood cells gave you a golden yellow colony. What microorganism causes that?
Strep pyrogenes.
Strep pyrogenes, which gives you what? Strep throat. And like I said, this is up until about 10, 12 years ago, this is how they used to diagnose strep throat. They do a culture on you, they rub it on blood agar, they give it 24, 48 hours, and they call you to let you know if you had strep throat or not. Now they have a dipstick test that works a lot faster. But up until about 10, 12 years ago, this is how you tested a patient for strep throat. Now, what is alpha hemolysis?
50%.
Yeah, this is usually a less pathogenic microorganism, so it only kills maybe 50% of the red blood cells. And since it only kills about 50% of the red blood cells, its colony is going to look green or maybe beige or even silver. And some of the organisms that give you alpha hemolysis are Staph aureus, Strep viridans, Strep pneumonia. They're typically found in the human throat, to be honest. In a young, healthy patient with a good immune system, they're usually not pathogenic. But if you had a geriatric patient who had a compromised immune system, then they could be pathogenic. But they usually give you alpha hemolysis. Now, what about gamma hemolysis, which is negative?
Gram negative.
Yeah, this would be if there's a gram negative. Basically, gram negatives don't kill red blood cells. Why? They don't release exotoxins. Remember, it's the digestive enzymes, it's the exotoxins from the gram positive bacteria. That's what's killing the red blood cells. Gram negatives don't have exotoxins. They have endotoxins. So you could rub E. coli on blood agar till the cows come home. You would never grow a single colony on it. You would just see kind of the grooves from your inoculation loop, basically. All right, so this is why blood agar is kind of the most common differential medium. different microorganisms will grow different colored colonies. Use it mainly to distinguish between gram-positive microorganisms. And honestly, this is how you diagnose strep throat up until about 10 years ago. Then some really smart person came up with the dipstick test, and that's what we use now. So, all right, questions? All right, we're going to stop there because this literally is your lab today. You guys are going to be throwing cultures on each other. Are you guys excited?
Yes.
Yeah, we'll see who throws up. No, I'm just joking. I'm teasing. Okay.
Are you though?
Mostly.
Has it ever happened?
90% teasing. How's that?
Has that ever happened in the lab?
Somebody has.
Not lately.
Oh.
Oh, yeah, not recently.